Today we're going to start with the cell, and there's going to be the cell organelles. Outcomes basically is going to be the functions and the features of the plasma membrane. So we talked about plasma membrane. We started talking about it in the last chapter, and that was on the phospholipid, right? You had a phospholipid bilayer, which is a phosphate, which is hydrophilic, and a lipid, which is a hydrophobic. And we said that any molecule that is lipid based is going to go through it because you have the lipids end on the middle. Now, the phosphate, which is the hydrophilic, is going to be on the outside of the cell, extracellular space, or on the inside of the cell, intracellular space. And that creates some sort of like a barrier between the inside of a cell compartment and the electrolytes in there, as well as the ones outside the cell. So that's the whole thing about the plasma membranes. We also talked about that in there, you're going to have proteins, and these proteins are going to be integrated in the membrane. You guys remember that, right? So these proteins are going to serve as channels. And these channels are going to have doors, and those doors, I'm going to call them gates. So whenever you see something that says channel or gate channels, it's just a protein that is embedded all the way through, and it has a door that you open and close it based on electricity, based on an ion, based on all the things. Okay, so that's pretty much it. And of course, attached to the plasma membrane on the outside, you have your receptors, right, or the antigens. So these receptors is what allows your cell to interact with the extracellular space. So what allows you guys to interact with the outside world? Your what? Your sensors, right? Your skin, your pain, touch, temperature, right? Your special senses, your vision, your hearing, your smell, all these things, correct? So those are pretty much receptors, let's put it that way, on the actual cell surface, which is going to detect what? Hydrogen, potassium, oxygen, glucose, amino acids. Is going to detect anything that is outside the cell. So that's what the receptors are for, right? To interact as well. So organelles and their specific functions, that's the next one that we're going to talk about, and that's going to be the majority of the content here. And then we're also going to talk about the functions of the cell nucleus, and you already know what is in the nucleus? DNA. And DNA is made out of what? What is the building block of DNA? Nucleic acids, right? And then, of course, they're going to make up your genetic code. And we're also going to talk about the role of DNA in the actual protein synthesis, which, to tell you the truth, is two places only. Protein synthesis takes place on the rough endoplasmic reticulum, or rough ER, or on the actual ribosomes. That's it. Nowhere else, right? So, cell structures and, of course, the cell function of the same, right? Because in the DNA, you have the genetic makeup, and that code is going to code for what? The cell membrane, the organelles inside, and the actual code of what they're going to be making. Or in this case, the proper word that the book uses is to be synthesizing, right? To making. So organelles. You can see here, organelles are mainly divided into two types. You have membranous organelles and non-membranous organelle. So the membranous organelle means that you have another layer or another membrane around the organelle. So extra way of protecting. Again, it's covered with that plasma membrane. So you have the ER, which is the endoplasmic reticulum. And what endoplasmic reticulum would that be? It would be both the smooth or the rough endoplasmic reticulum. So you know, ER has two of them. One is smooth and the other one is rough. Okay? So ER is smooth or rough. You can also, you're gonna see me writing like this. SER or RER. S for smooth, R for rough. So the second one is going to be the Golgi apparatus. So the ribosomes or these in the plasmic reticulum, one part of it is going to be synthesizing proteins and synthesizing lipids on the smooth. The Golgi apparatus, okay, that one is going to be able to do what? So this Golgi is the one who's going to assemble all these protein structures together. So let's say, for example, you want to make, I don't know, a hat. Saw that red hat. So if you want to make a hat, what are you going to do? You're going to need to get what? To put the hat together. Come on, guys. Okay, let's do shoes. Because it seems that hats, so you want to do a shoe. What do you need to put a shoe? To make it. You need rubber. What else? Glue. shoelaces, huh? Leather or cloth, right? And then who is going to be able to assemble this? It has to be someone who has the expertise to assemble all this rough product that you already have, correct? In this case, it will be, I don't know, China, Asia, Mexico, right? So almost nothing is made on US anymore. So the Golgi is the one who's gonna get this rough protein product and is gonna put it together to make sense of what it is. In other words, to make a structure like a shoe, a structure like a line, a structure like an alpha helix, right? A spiral, a structure like a beta plated sheet, so just a folded paper. So those are the structures. That's what the Golgi is going to do. Now the next one is going to be the lysosomes. And lysosomes are really good because these guys are able to do what? They are?
They're the trash cans?
They're the trash cans. In other words, pretty much they get all the food in there, like the food that gets into the cell, the lysosomes are going to be able to do what? Digest it. Anything that is rest or actually comes off of the actual organelles inside as they die, because inside the cell, things die, right? Like inside your body, you know things are dying? You guys know you're dying from the inside, right? Because not every single cell is live. Some cells do what? Die, and others replenish those cells, correct? So the lysosomes are going to be able to digest them as well. Okay, so those are the lysosomes. Peroxisomes are going to help you protect. If you have any bacteria or viral infection, these peroxisomes have hydrogen peroxide. You guys know what hydrogen peroxide is? Right, H2O2. Guys, what do you use hydrogen peroxide for? If you dye your hair, right, ladies or guys? I dyed mine once. So if you dye your hair, what happens? Well, you have to remove the color from the hair, correct? And you use that hydrogen peroxide, of course, at different levels. We're not talking about that hydrogen peroxide, right? We're talking about really small quantities. But the hydrogen peroxide becomes a radical, and this radical, what it does is just interacts with any molecule and destroys it. Okay? So as inside the peroxisomes, what are you going to find? Hydrogen peroxide. What is it going to do? It's going to kill, right, or destroy or deassemble anything that gets in contact with. Does that make sense? You also have a mitochondria. And you know the mitochondria, right? Mitochondria is the powerhouse of cells. Why we call it powerhouse? Because it creates ATP, right? So those are the membranous organelle. The non-membranous organelle are going to be the other ones. I'm not even going to list it, so you guys learn one and the other is the other. Does that make sense? So learn one, whichever one you think is easier, okay? And the other ones is the other one. Yep.
I know that like the smoothie R and the rough R is a different, like one has ribosomes and the other one doesn't. But like, is their function kind of the same thing?
No, it's a little different. We just started. We're gonna go into detail. So the main difference between the smooth and the rough is the smooth is gonna synthesize protein and detoxification. The rough ER is just gonna assemble proteins. But I'm gonna go in detail. So the next one is introduction to cells, and there are different types of cells, right? So we actually went into the actual organelles because organelles build what? Cells. I mean, I know we talked about the levels of organization, right? Which is the smallest one. It was the atom, then a molecule, then cell. But there is a link in between. That was the one that we just called the organelles, right? So now we're going to talk about the cells. So cells, there are two different types of cells in your body. You have your sex cells, okay? So your sex cells are known as the germ cells, and they're the cells that allow you to do what? Oh, sorry, not cells, but to reproduce, right? Reproduce means if you're a male, you're gonna create what? Spermatocytes. And if you're a female, you're gonna create what? An oocyte, right? Or an ovum. So once the spermatocyte enters the oocyte, then the egg is known as a zygote, and then you have, voila, fertilization, right? So fertilization is the process of creating a zygote. Now, the next type of cells, and you can see that the sex cells are the smallest of them all, right? They're the fewest, not the smallest, the fewest of them all. Now you have somatic cells. So somatic cells, soma means what? So soma means body, okay? And if soma means body, then those somatic cells means what? The rest of the body cells. So all other body cells, except, of course, sex cells, are what? Somatic cells. So if a question comes in and they ask you about a cell, you assume that is what? Somatic cell. If they ask you about a sperm or an egg, then you know that is what? A sex cell. Okay? Now, the process of cell division on this one is a little bit tricky because it's different from one and the other one. So what is the cell division here? For sex cells, right, the cell division will be what? Mitosis or meiosis? Meiosis. For somatic cells, it's going to be what? Mitosis. And I believe we talked about it in the lab, right? If you're one of those students. Now, what is the main difference? Well, mitosis and meiosis go through much of the same cycle of phases, right? Which is the P, mat, C. I'm going to explain a little bit in what it is. So P mat C means what? P for prophase, M for metaphase, A for T for C for cytokinesis. And the same process occurs on mitosis. Okay? The only difference is that in meiosis, the PMAD happens not once, but happens what? Twice. Everybody got it? So on meiosis, this process happens once and twice. In mitosis, the process only happens once. I'm not going to go into a lot of detail. That's just the intro of it. So you know the difference between the two. Okay? All right, let me just go back. So it means that every single time that you create a spermatocyte or an oocyte, what process should you go through? Mitosis or meiosis? Meiosis, right? How many cycles? One or two? Twice, right? To the PMAD cycle. Okay, awesome. So what are the principal parts of the cell model? And again, this is just basic, but the cell is composed of a what? Cell membrane, or also known as the plasma membrane. So those are two synonyms right there, or plasmalemma. Oh my God. 3 words which means the what? The same thing. So cell membrane, plasma membrane, or plasmalemma all means the cell membrane, all of it. Now, the next thing is going to be the organelles, right? The organelles that we start talking about, they're membranous or non-membranous organelle, the main one, that main division between those. You also have a cytosol, and a cytosol is the fluid inside the cell. You also have a nucleus, which is the nucleus where you have your genetic material, right? That we talked about, the DNA, that is made out of nucleic acids. Now, we also have other inclusions like what? Glycogen, lipid, pigments, etc. You need food, correct? Your cell needs it as well. That's why you're eating. So that food is somehow going to get into the individual cells of all your body, right? And those are some sort of the examples of inclusions, okay, inside the cell, which of course your cell is going to be able to use, right? We talked about the molecule of glucose that is going to be able to use to create what? ATP, right? In the mitochondria. So mitochondria most of the time uses what? Glucose, and converts it into a molecule of ATP. So the cytosol, the organelles, all these inclusions, right, is part of the what? The cytoplasm. So inside the cell, there are 2 main areas that your books focus on, and that is cytoplasm and nucleus. So there are going to be cell processes that will take place either on the cytoplasm, which is outside the nucleus, inside the cell, but outside the nucleus, right? Or within the nucleus. Everybody got that? Guys? So the cytoplasm is just the space outside. So now we're going to go to each one of those organelles, and that's just a summary in one single page. The rest of the chapter is going to be talking about this picture right here. So let's start with one. First one, we're going to talk about the plasma membrane, and that is the cell membrane, right? And if you look there, the plasma membrane or the cell membrane, it is made up of what? Phospholipid, what? Bilayer. So it's a phospholipid bilayer. So this is a two lipid bilayer, right? Or I'm sorry, a phospholipid bilayer. I should put the phosphate right here. Let me get something smaller. All right. So the phospholipid bilayer. Now, this lipid bilayer that you have here, inside of it, you have the proteins. So remember the membrane proteins that we talked about? So these proteins can come into different ways. So proteins can be either embedded, which is the one that you have here, right? Or the proteins can be what? Superficial. Superficial or integral proteins. Integral means embedded. So integral proteins are these ones right here. Why are they called integral? Because they're integrated in the whole what? Phospholipid bilayer. It goes through the whole span, right? Now, the surface is going to be either on the inside or they can be what? Found on the outside, okay? So there are two main type of proteins that are going to be part of this membrane of the phospholipid bilayer, right? Integral or superficial. Now, the superficials are going to be used mainly to bind to what? To cell, and that little y means what? To cell receptors. Okay? Remember the receptors that we talked about at the beginning? That's the way that the cell interacts with the outside. So the next one that we're going to talk about is going to be the integral protein. So integrals, right? These are going to be known as what? We talked about it too. These are going to be called gated channels. And the gated channels can come into different forms. The gated channels, they have a gate, and all it is that they're talking about is this, right? You can see that the channel is here, correct? There's a channel created there in the middle. The gate is just that it has a door or a trap door. The door can be what? Closed. Here, you can see it that it's closed. Or the door can be what? Open. If the door is open, then any ions, right, any electrolytes, anything can actually go through as long as it fits through that channel. Like if I put the door open here in the classroom, right, anybody can go through, in or out, as long as you're able to fit through it. Does that make sense? So these gated channels can be open, these gates, right, or can be open the doors, either with a electrical stimuli. What is an electrical stimuli? Have you ever been got electrocuted? Right? Have you ever got like a little bit of 12 voltage, right, on you? So that is electricity. It means a small charge that will activate the door to open. Like probably you guys have it in your house or your condominium, right? You click on the remote control activates a motor, and that motor has electricity, and the motor just moves the door open or the gate open. That's what it's doing, right? So that's the electrical one. Then you also have a chemical. So these are the different ways that these channels can be opened. Chemical means what? Chemistry, right? Chemistry means an ion. Means sodium, potassium, chloride, magnesium, right? That is going to bind to a receptor. Let's say it's going to bind to a receptor, or we can just draw another receptor. So let's say this is sodium and this is potassium, right? It's going to bind to receptors, and these receptors will change the actual way that the door will open, okay? So that is the chemical. And finally, we have a mechanical. What is a mechanical? Mechanical is a physical event. So I want you to touch yourselves. Touch it. That is mechanical. It means there's gotta be a actual movement of the what? Of the cell membrane, because this is embedded on the cell membrane. So when you hit a wall, right, the cells are being doing what? Moving mechanically. So these can elicit or trigger ions or gate ions, right, to do what? To open. Does that make sense? All right. So these are the ways that the channels can open. Any questions? So when we're talking about protein synthesis, please refer back to these things. Most of the structures or the organelles inside the cell are made out of what? Proteins. So almost all the organelles, the majority of its composition of organelles is going to be made out of what? Proteins. And proteins has an overall what charge? What is the charge of a protein? Negative. Just please know that inside of the cell is a big what? Negative charge. So if the inside of a cell is overall negative charge, the outside of a cell, hopefully, is a positive charge. So if you think about this one, the negative inside is because of the protein, and the positive outside is because of the ions, mainly sodium. So there's an electrical charge difference, correct? And whenever you change polarity from positive to negative, what do you get? You're thinking about biology? I'm thinking about real life. What is this?
Battery?
Right. So this is a battery. A battery has a positive end and a negative end. Whenever you put the negative lead to the positive lead, what is here? What is flowing here? Electricity, right? That's the same thing that is happening from here to here. When there is a difference in charges inside and outside the cell, there is a electrical current that is surrounding each one of your cells. Okay? So, correct me if I'm wrong, there's something known as an aura and a chakra. You guys ever heard that before? So your chakra is what? The chakra is actually the outside energy that surrounds your body, as far as I know. I'm not sure 100%. But definitely, people say, oh, these people are crazy. You know, that's stupid. It's not. It's actually true if you think about this. If every single cell in your body has a difference in charges, positive and negative, that means that when you're walking through a place, you're leaving behind what? A charge around your body. So there is a charge around your body. That's why sometimes when we put a lot of friction, something, and we touch somebody, we're just gonna get what? Electricity. Have you ever done that before? Yeah. So there is electricity around us. Based on what we do, electricity changes. Based on how the cell interacts, electricity changes around or within the cell. Does that make sense? All right. The take-home thing is the inside is negative, the outside is positive. Awesome. So let's go to the next one. So where we have number 2. Number 2 is your mitochondria. Number 2 is your mitochondria. So mitochondria says here, organelle in which the energy is extracted. What does that even mean? That means that the main energy or the main fuel, right, molecule that your mitochondria has a predilection for is going to be what molecule? What molecule will that be, guys? I said it a couple times. That molecule that comes in here, no, he produces ATP, but one molecule is required so you can extract glucose. So where do you get glucose from? Food, right? Glucose is a monosaccharide, right? So as you get the glucose, glucose goes inside the cell, right? How do the food get inside the cell? There is either through a channel, right? But anything that enters the cell, almost everything that enters the cell, not everything, but almost everything that enters the cell, in example of macromolecules, glucose is a macromolecule. That process is known as endo, which, what does that endo means? Inside, and then cyto means cell, and then cytosis, okay? So the process is known as endocytosis. So all macromolecules, humongous molecules, you require a big door, right? Or a vesicle that is gonna come inside and bring it over. If your molecule is a micro, not a macro, this is the example of macro. If we have a micromolecule, and I believe we talked about it, what is a micromolecule? Water, oxygen, CO2, right? These are tiny molecules, right? They are able to go freely through the cell membrane. Why? because they're really small and they can go in between each one of the phospholipid bilayers. So if you put your hands like so, right, there's always going to be a gap between the two hands, right? If you're really small, what can you do? You can just go through it. Does that make sense? And that's what happens to the small molecules. Small molecules can actually slide through each or in between each phospholipid bilayer. So you have, now, we just talked about the process of endocytosis, which is mostly used for what? Macromolecules, okay? We also talked about the micromolecules that are really tiny molecules and they can go freely inside the cell membrane. They don't have to be hydrophilic or hydrophobic. They're so small that does not affect them. Why? Because the hydrophilic and hydrophobic affects molecules that are really big. Okay? Does that make sense? Now, if you are a macromolecule, and you are a lipid base, so this is a sugar, but if you are a lipid, can you go freely through the cell membrane without using a door? What's the answer? Your cell membrane is made of a phospholipid bilayer, right? So I'm just trying to draw in these things right here. Hopefully you guys get it. That means that the middle here is a lipid and the outside is the phosphorus. So what does that tell you? That anything that is lipid soluble, which is most of the span of the bilayer, it is able to do what? Guys, most of the lipid molecules are able to go freely through. Why? Because most of the bilayer is made out of what? Lipid. So macromolecules, as long as they're lipid soluble or lipid base, hydrophobic, you guys get all those? So lipid soluble or lipid base or hydrophobic, which is all the same, right? They're able to go through the phospholipid bilayer freely. They don't have to go through the actual door, which is in this case is the gated channel, or the process of endocytosis. They can just go freely. And for this, most of the time, we're talking about what? These lipid-based molecules, we're really referring to hormones, which is another event on 2086. Okay? Ever seen movies? And ghosts can go through walls? Why? Well, normally you're made out of what? Atoms, right? The simplest atoms. If your atoms, which how they make composed of the organelles and cells, they vibrate. We all have a resonance, right? It's all about sound. Life is all about sound. If you have an electrical resonance, which is vibration of these atoms, is what creates a physical, palpable what? Hard matter. If your molecules are not vibrating as fast and they decrease the vibration sequence, right? You can actually go through the cell or through the matter. Does that make sense? Air. Does air has matter? Yeah. Can you go through air? Yeah, look, I'm doing it right now. So if you're able to slow down the vibration of the atoms, you will slow down the vibration, as a theory, of the cells in your body, and you're able to just walk through things. Right? Because it's all about the frequency. Again, this is just anatomy. I'm not going to go into that, but that's, bringing in the stuff from the real world into this. Okay? All right, so let's go to the next one. Let me erase this, so I don't want to be a lot busy. All right? Sorry, do you guys want to take pictures? I'm going to upload it. All right. So that we did the mitochondria, right? So whenever you use a molecule of glucose, what do you get? So glucose comes in, right? What do you get? Oh, what do you need? You need oxygen, right? Because that's what we call cellular respiration, right? Because the cell is breathing, we are breathing, right? That's the reason why we need oxygen. And then as a side effect, what do we get? Side product. CO2, ATP, and hopefully we get something else. Water. Okay? For almost all cellular reactions, this side product is almost all the time constant. You almost all the time get water and CO2. Okay? So that's number 2. The next one that we're going to talk about here is your cytoplasm. I think that's number 12, but I don't know. Anyway, so cytoplasm. So cytoplasm, it says a semi-fluid matrix, right? Means it's not purely water, but it's a watery, gelatinous kind of like conformation, right? So the cytoplasm contains in there what? Your nucleus and all the rest of the organelles. So cytoplasm is the inside of a cell. Now there's a difference between a cytoplasm and versus cytosol. What is the difference between cytoplasm and cytosol? The cytosol is the watery compartment. That was supposed to be an H2O. The watery compartment inside the cytoplasm. So it's like a gelatinous medium, right? The watery part of it is the cytosol. Almost all chemical reactions takes place where? On the cytoplasm or cytosol. So far, good? Right, so you need to know that difference. So I have no number 3. Should we go to number 4? Or should we just, number 4. So this cell membrane, right, is going to have a shape. Like you all have different shapes, right? You may look the same, but you all have different shapes. Now, in able to give the shape of a cell, and what am I talking about the shape? You see the shape here, right? There's an indentation here, there's another indentation there, there's a rounded area there. So not all cells are created equal, right? Because your cells are what? Since we're talking about cells, let's hit it. So cells, 3D shape. The cell's 3D shape means what? What are the different type of cells that you already have? It was covered in the lab. What are the different type of cells that you have? You should have all had this. What are different type of cells? No. The shape, the 3D shape of the cells. Cuboidal. Columnar. One more. The one that looks like a fried egg. Squamous. Okay. So squamous, all right. Cuboidal, columnar, and squamous. All these are different type of cell shape. What gives that cell shape? Well, that is your what? Cytoskeleton. So cytoskeleton is what gives the different type of shape. Now, what structures are part of this cytoskeleton? Now, cytoskeleton, the main thing that it does is, apart from giving the shape of the cell, you can see here, it also supports what? Organelles, and plays a role in cell motion, means cell division, cellular movement. Imagine that you're alive without roads. You have a car, but there are no roads. How do you get to school? It may take you some time, right? So the roads, in this case, inside the cell, is what allows the organelle to move from point A to point B. They're not static, right? Imagine this is a city inside, and things are moving. So for them to move, they require some sort of pavement, right? This is some sort of railroads. So these are known as what? Microtubules. Molecules present on the cytoplasm, cilia, or flagella. In this case, these structures, the microtubules or intermediate filaments or actin filaments, right, allows for things to move inside the actual cell. Everybody got that? Now, we're gonna talk about those in the whole semester. So the other ones for the cytoskeleton, not only microtubules, but you also have what? Intermediate filaments. Okay? What they do is they provide support and strength, and this is actually for the fibers. Okay? Have you guys heard like the fiber, protein fibers? Okay? So what makes that thing structure harden, right? Or the muscle to be get like hard. So actin filaments is the other one, and this we're gonna see actually on muscle interactions. You're gonna have actin and myosin filaments, and they allow for the muscles to do what? Contract and relax. Okay, that's pretty much what they do. So those are structures of the cytoskeleton and function of it. The next one we're going to talk about is your nucleus. So what about the nucleus? The nucleus is the command center, so everything happens. In the nucleus, you're going to have what inside the nucleus? In the nucleus, you're going to have DNA. DNA is going to be made out of what type of sugar? Sugar? Deoxyribose. Guys, don't forget what you already learned, right? And then this DNA is assembled by a group of what? Nucleic acids, right? So the DNA molecule is composed of what structures? A double helix. What else? Sugars. Then, and a phosphate backbone, right? Awesome. So inside this DNA, right, this assembled structure is your genetic material. Is the one who's going to code and is going to allow for all these things to be made, to be created, right, to be exported, or to be used. All the information is there. It's like the brains, all the stuff. So inside the nucleus, there's a smaller structure, and that is known as the what? Nucleolus. Now, even though the nucleolus is inside the nucleus, the DNA is in the nucleus, but in the nucleolus, all you have is genetic material, right? DNA, but that only does one role, and that role is to make ribosomes. Why would you spend a majority of a nucleus for ribosomes synthesis? That means that if you take space from your own DNA to make a specific organelle, that means that that organelle must be extremely what? Important from the life of your cells. Does that make sense? Think about this. If as you're aging, right, what allows to keep ourselves in a good healthy state, in a good looking state, is the amount of genetic material that is read and expressed inside your cells. If your cells start losing ribosomes, or you don't make any more proteins, or you don't have sufficient mitochondria, what do you think is going to happen to your cells, to your skin, to your tissues, to your organ, to everything in your body? It's going to start to die off. And that is the moment that you start to become what? Sick. Well, I'm starting about getting older, right? You get sicker, weaker, right? Because that information is not read anymore within the DNA. So ribosomes, the nucleolus is just to make ribosomes. What is the whole purpose of the ribosomes, right? The ribosomes is to make proteins, as sites of protein synthesis. So whenever you see a ribosome, you are going to make what? Protein. What can we make out of protein? We making what? What do you make out of protein? Muscles. What else? Hey, let me write this down. Let's do a list, because I'm gonna give you a few, but let's do it over. So we make muscles. What else? Enzymes. We talked about enzymes already. What else? Muscles, enzymes, tissues. We just talked about something. Huh? Fat, no. Inside the cell, most of the structures are made out of what? Organelles, right? Did I say most of them are made out of protein? So guess what? Organelles are also made mostly out of protein. That means that, is protein important? Well, based on that list, I think so. So that's why it takes so much of a space out of there. Okay? So ribosomes, where do you see these ribosomes? Well, ribosomes can be seen, if you look at it right here, they're bound, right? They're just fixed to this wall. What is that wall? Let's see. Where is that wall in the picture? The wall, and if you look at it, let's go all the way here, and it's known as the rough endoplasmic reticulum, the RER. Remember RER? So this rough endoplasmic reticulum, RER, is going to allow it to do what? Protein synthesis, the same thing as the ribosome, because that's what they're doing. Now this protein synthesis in here is going to be used for what? That protein is for only exportation. Everybody got that? Now, the ribosomes, and you're going to see all the ribosomes. Let's see, we're here. Where are the other ribosomes? I guess they don't put them anymore, but anyway. Do you see this one right here? Yeah. So this one, we call it a free ribosome. Okay? Why are they free? Because they're not bound. What are they bound? Because they're attached to the wall right here. You see there's a wall and they're attached? So when they are actually bound, is that they actually are attached to the endoplasmic reticulum. Once they're attached, we call them what? Rough endoplasmic reticulum. If they're not attached to anything, we call them what? Free or unbound. Okay? Like if you have a significant other, you're means you're what? Bound to that person, right? If you're looking for a partner, you are available. We call them free. So that's pretty much how it is. So they are free. But the main difference is, what is the function of the free ribosomes versus the rough ER? So I'm going to put them here. So free ribosomes, what is the main function? Guys, what is the function of the ribosome, regardless whether free or bound? To synthesize protein, right? So protein synthesis is the same thing. The only difference is that this one is not for export, this is for what? Import, but we're not importing anything, it's just internal internal use. Why are you talking about, what do you mean internal use? Guys, I believe when we talked about ribosomes, we said that it can make muscle, enzymes, and tissues, right? Are these import or export? You're going to take it out of the cell, right? Because do you make muscles inside the cell? No. Because you got cells here and then there are muscles there. So this is for export. So that means that this portion comes from what? The RER. Everybody got that? Now to make organelles, do you really need to export them out? No, organelles are within the cell. So that is for internal use. So that is your free ribosomes. Everybody got that? If you're following along the way I'm separating it, it's going to take you a lot easier route to go through this chapter because they kind of make it a little complicated. Any questions before I move forward? No? All right. So the next one that we're going to talk about is the smooth endoplasmic reticulum. The smooth endoplasmic reticulum is basically for what? Let me open that up. And it says carbohydrates and lipids. It says manufacture. It means you're going to create what? You're going to build, you're going to synthesize what in there? Carbs and lipids. What carbs? What carbs do you make in your body? How do you store carbs? What is the way of storing carbs? We talked about it. Glycogen. So the carb that they're talking about is what? Glycogen. So glycogen synthesis. The glycogen that is stored in your liver. So what are you going to have a lot of smooth endoplasmic reticulum? In your liver cells. Does that make sense? Because that's where you store most of the glycogen. Where else are you going to have a lot of smooth endoplasmic reticulum? Where else do you store glycogen in your body? In your muscles, right? So you're going to have a lot of smooth endoplasmic reticulum there. It means that this organelle will be predominantly increased in both the liver cells and or the muscle cells. Okay? Any questions? Awesome. Now the next one is lipids. We're talking about hormones, sex hormones, right? How many of you like to have more of what you already got? More estrogen, more testosterone? No? You okay with what you got? All right. I want to try it. All right, So more estrogen, more testosterone, these are means that are sex hormones. So where are these sex hormones built from? If you think about it, sex hormones, they're mainly made out of what? We said that sex hormones are made out of lipids. We call them fats, right? So if your sex hormones are made of fat, right? And you said, oh no, but my sex hormones, what do you make your estrogen, ladies? Where in your body? Ladies? Ovaries, right? And then if you're a guy, what do you make your sex cells, guys? I mean, your testosterone, what do you make them? On your testes. Awesome. So, but that means what? Testes? This is just talking about like the whole organ. But now, this is actually inside the cell, in the actual organelle that is doing that. So in your gonads, you're going to have high amounts of what? Smooth endoplasmic reticulum, because you are building, right? Sex hormones. Does that make sense? So that's the smooth endoplasmic reticulum. So not only on the liver or muscle, but as well in your gonads. Okay? Whenever you take exogenous amount of hormones, let's say you take too much testosterone or estrogen, what happens? If I give you $10,000 a week, would you be willing to work?
Yes.
Most people say yes. I give you $10,000 a week. You want to work?
No.
You don't have to. No. That means that if you're giving exogenous hormones into the smooth endoplasmic reticulum, will they work? Will they produce more testosterone or estrogen? No. So that means that you are going to be arresting or stopping the production or synthesis of lipids in where? In your gonads. It means in your testes and your ovaries. So if you're a guy and you're a bodybuilder, why? If you take a lot of growth hormone or testosterone, what does that lead to? On the long run, your testes will get what? Smaller. Why are they getting smaller? Because the smooth endoplasmic reticulum in there don't need to work. So that organelle for sure is going to be arrested. Now, once you stop the testosterone, that's why you don't stop testosterone, you do it in cycles, right? When they do bodybuilding, and you take them off in cycle as well, you decrease in dosage. So that's why they're able to work. But if you take them completely off, it's like the corticosteroids, you shock your whole system, so they won't work for a while, if they ever work. Okay? So you have to decrease them, right, until you can take them off. So this is called weaning off. Okay, so the next one is your peroxisomes. So what do peroxisomes do? So peroxisomes, they have hydrogen peroxide inside them. We kind of did talk about them. So they contains enzymes, and these enzymes are going to be what? The enzymes that allow you to detoxify. Okay, so the hydrogen peroxide, all right, these enzymes create hydrogen peroxide and they detoxify. Have you ever been toxified? If you drink alcohol, if you smoke anything, if you eat anything out of a bag, any processed food, if you take any medication, right? Even the vitamins that you take, they all have what? Toxins, okay? Because most of the stuff that you take in medications is not the active ingredient, is most of it is what? The byproduct. People call it the talcum powder, right? Which is not drugs, but anyway, it's just the actual product that makes things visible so you can ingest. But most of it is an inactive product. But it's a toxin. It works as a toxin because your body has to do what? Get rid of it. So detoxifying happens mostly where in your body? Everywhere. But there are two main places. The number one is your what? Liver. So if you're drinking a lot of alcohol and you're taking a lot of drugs, if you're eating a lot of garbage food, a lot of processed food, that is all who's taking the hit? Your liver. Follows by who? Your kidneys. Liver takes hit on everything that is lipid soluble, right? Anything that is lipid is going to break it down. But the liver converts it in from lipid to actually what? Water soluble, so now your kidneys can pee them out. Okay? So those are the two main detoxifying portions in your body. So peroxisomes, organelles will be highly numberable in there. The next one is what? Centrioles. Centrioles is a complex assembly of microtubules that occurs in pairs. These things, I call them churros. They look like churros to me, or macaronis. Okay? So these centrioles, all they do is from within them, they're able to create inside of them microtubules. But the main function is to use as what? Cell division and communication. Mainly for cell division and communication. If you remove the centrioles of a cell, can the cell go through mitosis or meiosis?
No.
Impossible. That means that once the cell dies, what happens? It goes bye-bye, you don't get another cell. The thing why we are alive, where all cells are constantly dying, but they're actually constantly what? Reproducing, replicating, right? So that is because mainly of this organelle right here. I mean, all of them does a great function, but mainly on this one because they will be able to separate. And last but not least is secretory vesicles. Anything that, oh no, there's one more. Anything that you don't want, what are you gonna do? Excrete it. How are you gonna excrete it? The process of excretion is known as what? The one for inside the cells called what? Endocytosis. And the excretion is what? Exocytosis. Everybody got that? So endo, inside. Exo, outside. So these secretory vesicles, they don't only excrete the garbage or whatever byproducts you don't want, but actually says plasma membrane, so they fuse with the plasma membrane, but release material to be secreted from the cell. So that quite means garbage. Why? Because as you make the proteins on the rough endoplasmic reticulum, ready? So DNA, let's do this. Let's start number one, DNA. DNA sends the genetic material, right? Sends the material outside on the rough endoplasmic reticulum, correct? This genetic material is known as messenger RNA. Why we call it messenger? Because it releases what? A message from DNA to RNA. So message to whom? To RNA. Now, this message is going to be read by the ribosomes inside the rough ER, right? These ribosomes, what are they going to do? They're going to assemble what? Amino acids together, correct? As you assemble the amino acids together, you're going to put them on a secretory vesicles, right? That will go into the what? This one. This one means what? This is your Golgi. So once it goes inside the Golgi apparatus, it's going to assemble. Remember the shoelaces, the fiber, the things that we got to make the shoe, right? The Golgi is the one who's going to assemble all these proteins together, a bunch of protein that is sent into one protein that makes sense. Once that is put together, then the Golgi is going to send a secretory, or in this case, a lysosome as well, but mainly is a secretory vesicle to get what? The product outside the cell for exocytosis. Everybody got that? Yes? All right. So once you get all this group of amino acids, you don't get one, you get like multiple amount of groups that are sent over here, right? Into all this, they're all in there. So all these amino acids are sent over, right? A vesicle that goes into your Golgi. This is in the cis configuration. I'm not gonna go in details. We're gonna talk about that later. And then it goes out of the Golgi to the transphase. The Golgi is gonna put them all together, assemble it into something that makes sense, okay? And then it's gonna go into a secretory vesicle that is going to then allow for things to get out of the cell. To make what? I don't know, whatever it is that you wanna make, right? A muscle, an enzyme, a tissue, whatever you wanna use that protein for, okay? So I guess that's it for today, guys.